Our next presentation in session 1 is accelerated drug development and precision pharmacotherapy using DeepPumas. And this is presented by Dr. Niklas Korsbo. He's the lead developer and scientist at DeepPumas and Pumas AI. Thank you.
Hi everyone, I'm here to talk about accelerated drug development and precision pharmacotherapy using DeepPumas. My name is Niklas Korsbo. I'm a scientist at Pumas AI, where I lead the development of DeepPumas. And DeepPumas is our way of bridging the gap between machine learning and scientific modeling using scientific machine learning. And in machine learning, it's fantastically good at doing automatic model discovery of finding unintuitive relationships and handling complex data. But it can be hard to utilize your scientific understanding in such models. It tends to require big data, and the results can be quite inscrutable. In scientific modeling, such as, for example, a PK/PD model, there you can really encode your scientific understanding into the model, and that makes the model data efficient, it makes it transparent and interpretable, and you can run counterfactual simulations for example, dosing optimizations. But creating them is labor intensive, and it's easy to miss some unintuitive relationships that are important. And it's also hard to utilize complex data. So with DeepPumas, we make simple and effective utilization of both knowledge and data. So you might have some data from measured outcomes, you might have some images, some omics, but you also might have some understanding about the disease that you're studying. So you might know some of the molecular interactions and some of the drug properties. And so we want to be able to combine all of this information, all of this knowledge into the same model in order to make the best possible prediction. And this idea is applicable across the entire modeling space in the pharmaceutical industry. But today we'll be using it on a case study of an ongoing case study on oncology. And so here we have some synthetic data where we have access to tumor sizes over time for the different patients. And we also have access to survival data. So not all patients survived for the entire 24 months of this simulated clinical trial. So from this data set, we can ask ourselves, what is driving the tumor dynamics? And what effect does the drug have? And how does tumor size affect the survival of the patients? And so I should add that throughout the presentation here, we've been using a training data set of 200 patients. So it's not a huge data set. In order to move forward, I want to have a quick reminder of what is a neural network. So a neural network is an information processing mechanism that's loosely based on the idea of neurons, where every neuron takes a bunch of inputs, it processes that input with some nonlinear function, and it can produce an output. And individually they're quite simple, but if you stitch them together in a network, that becomes quite capable of processing information. Mathematically, most neural networks are just functions, and this means that neural networks should be usable anywhere where you would use a mathematical function. Also, they are universal approximators, meaning that they can approximate any functional form. The form they take on is determined by their parameters, and the fitting of those parameters could be linked to, for example, patient outcomes. And this means that you can really use data to automatically discover relationships that are important for the disease progression and for the treatment. Now, we'll have a look at a neural embedded tumor size model where we ask ourselves, how do these tumors grow and why is there differences between different patients? And so for this, we define a dynamical system where we say that there's drug coming into a central compartment where it eventually decays from. We then take a leaf out of this paper, Wang et al. 2009, where if you're reading between the lines a little bit, you can say that they have a model where the tumor size is split into two. One part which is responsive to the drug and one part which is unresponsive to the drug. And this seems to yield some good results. So we'll take that and we'll make that assumption ourselves. But here comes the problem. So what equations should we use in order to describe the tumor sizes over time? And I would say that there's no good answer here. There's nothing in biochemistry or in biomechanics that would tell me how I should model the tumor size growth. And so here we would use neural networks. So we define one neural network for each one of these variables. And I can define what input these neural networks take. So I say that the responsive tumor size, it takes the drug as an input, it takes the tumor size itself, some random effects, and some processed covariates, and we'll get to that in a second. And the unresponsive tumor size, it simply does not see the drug come in. Like, the drug is not part of the input, so it can't be used. And instead, you only have it that it depends on the tumor size itself, some random effect, and some processed covariates. All right, and so this enabled me to then say that I don't really know the functional form that are important here, but I know the structure and the information structure of this model. So I know which entities might affect what, right? And this is also information that we're encoding into the model, and that encoding of that information makes the modeling quite data efficient. So here we also have some parameters, and this is a nonlinear mixed effect model where we have some typical parameters, so typical values that are shared across all of the patients in the population. We have some random effects that is accounting for the sort of unpredictable or unexplainable heterogeneity between different patients. And then we have some covariates. But here I don't actually know how to process these covariates. I don't know what they should be used for. And so I will again use a neural network to process all of those. And the neural network gives me this vector of covariate features that I can use throughout the rest of the model. And so here, for example, we have one for estimating the volume parameter. And so the idea here is that now that when you do the fitting, the neural network has to then figure out how to best utilize the combination of all of these covariates, some nonlinear combination of all of them, in order to best predict or individualize the volume parameter for the different patients. And then we use a similar thing for across the entire model here. You see it in this initial tumor size, and we see it in the dynamical systems. And, in this case, we've been using numerical covariates, but we've also been using images in other projects. So you can really use complex covariates in this kind of modeling. For completeness, I'm listing some random effects that we have, and we have that there's some observational noise where we say that the tumor size is the sum of the responsive and unresponsive tumor size with some additive noise to it. And also important is that the neural network parameters are just fixed effects in this model. So that means that everything in here, all of the parameters of this model, including the parameters governing this machine learning, they can all be tuned and fitted together when you're fitting towards the patient outcomes. So we do that, and then we can make some predictions. As from here, we're making predictions based on baseline data. So this is the covariates that we have available, and it's the first measurement of the tumor size. And so we use that data, the data we have on day one. And we predict onto test data, so we've never used this during training. And here we see that the DeepPumas model has quite accurately identified how the tumors evolve over time and how to relate to that. And it also identified how to use the covariates to individualize these predictions. Next up, what I'm most interested in here is not the tumor size, but rather survival. And so one way of modeling survival is to model the cumulative hazard, which is integrating the hazard over time. And the cumulative hazard has a connection to the survival probability of the patients. And the tricky part of such a model is to determine what is the hazard. And one way of doing this is to use an exponential survival model where you say that the hazard is a constant, and then you get exponential survival. You could say that the hazard is you can have a Weibull model where the hazard is a time-dependent function. And I would say that it's actually a little bit hard to say what is correct here, and it's quite arbitrary. So here I would just use a neural network that takes time as an input, and it gives me hazard as an output. And I'm fitting this with our data, and we see that the predictions that we can make here is very well aligned with the training data. And if we simulate the overall survival from this fitted model, we find that the distribution of overall survival matches quite nicely with the training data as well as with the test data. And here we can also ask ourselves, what's the relationship then between hazard and time? And here we see that the hazard starts off high, and then it goes down a bit, and then it goes up again. And this might be then related to, in the data we had that the tumor sizes start high, and then they shrink, and then they grow again. So that might be related. This kind of modeling that we've done here, is quick and it's universal in that the same model could be applied to all sorts of data. It's fine with only survival data. But there's a risk of overfitting when you use explicit time like this. And using explicit time also has a drawback in that time doesn't really do anything in your body. So you wouldn't have a mechanism in here, which means that you can't really run counterfactual simulations. So something I'd be more interested in is to see if there's a relationship between tumor size and survival. And so rather than using time as a parameter or as an input to the hazard, I'm going to use the tumor size because we just modeled the tumor size before, right? So now the question is, what function should relate the tumor size to the hazard? And again, I'm going to use a neural network to automatically discover this. And fitting this, we see that the resulting overall survival that we can predict, it matches really nicely with both the training data and the test data. And we can ask ourselves, what's then the relationship between tumor size and hazard? And because we're using synthetic data, I have access to the ground truth, and so we can compare with that. And we find that the DeepPumas model quite accurately predicts what the relationship between tumor size and hazard should be. And so now we really have a way of predicting individualized tumor size over time. And we have a relationship between tumor size and the hazard, which means we have a relationship between tumor size and survival. And this means that we can really go beyond overall survival and into patient-specific survival predictions. So here we're predicting the probability of survival, and here you see 6 different patients, and the way that you will read this is to say that the probability of this patient here surviving until month 12 looks like it's around 75%. And the probability of them surviving until month 24 looks like it's around 20%. Right? And so here we're making individualized predictions of patient survival based on their baseline data, the data we have available when they first come into clinic. And we can also refine this as the patient comes in. So rather than simply relying on the the baseline data that we have available, we can take the data as the patient comes into the clinic and we measure the tumor size over and over again. We can use that data to inform our model and revise it and make better and better predictions. And when we do that, we don't only improve the tumor size predictions, but we also improve our estimated survival probability. Another thing that we can do is to have patient-level dosing guidance. So here we take the same patient 3 times and we ask ourselves, what will be the probable consequences of not dosing the patient at all? And here we'd see that, okay, so we're expecting the tumors to keep on growing and the survival to go down really quickly. And if we do give them drug, then the tumor size will stay lower, it'll still grow eventually, and the survival will be much better for longer. With an even higher drug, then the tumor size will be even smaller and the patient will survive even better. Right? And so I think that one of the advantages of being able to do this is that, like, if you have a model which can quite accurately then predict survival probability on this individualized level, then you can ask yourself questions such as, what is the consequence of making a different dosing decision? So what is the consequence between picking one dose over another? And also, what is the consequence of, like, this patient taking a vacation, going to Barbados for a month and not getting any treatment during that time? What is the consequence in terms of expected time of life? And having access to that makes it easier to reason about these decisions and to make informed decisions that might be appropriate for this patient. And so here, DeepPumas has identified tumor dynamics We've identified the drug effect and the covariate effects, as well as the relationship between tumor size and survival. And with this, we've been able to make predictions, and we can continually improve the predictions as data comes in. And we can also quantify survival effects from different treatment options. And this kind of modeling can be used with other kinds of biomarkers, covariates, and time to event observations. And we've used it outside of oncology as well. So we've used it in ophthalmology and even epidemiology. And so this is a fairly powerful approach, I think, and we're quite excited about this. So with this, I would like to thank everyone I have worked with here. It's not a solo mission. And I would like to thank you for listening. Thanks. Bye.